2. The Crisis in Forensics. In fact, it was the pursuit of a good story that has led me to the world of wrongful convictions, said John Grisham, the best-selling writer and attorney, speaking in the U.S. Senate. Grisham stumbled into the world of forensics when he happened to read the New York Times obituary of a man named Ron Williamson. We were the same age. We both dreamed of being Major League Baseball players. We both grew up in small towns in the Bible Belt, and we both came from the same religious backgrounds. Yet Ron was convicted of a rape and a murder he did not commit, was sent to death row, went insane, and came within 5 days of being executed before receiving a miracle reprieve. Grisham began to write his first nonfiction book, The Innocent Man, to tell that story. The case provided his introduction to wrongful convictions and faulty forensics. In that case, the prosecutors had put forensic experts on the stand who gave faulty testimony about hair comparisons and blood typing. Grisham recalled, "The most damaging testimony against Ron came from an expert, an analyst with the Oklahoma Bureau of Investigation. This expert testified that there were 17 scalp and pubic hairs taken from the crime scene, and that the hairs were microscopically consistent with Williamson's. 11 years later, each of those same hairs was DNA tested, and none came from Williamson, who was exonerated but died 5 years later of liver failure." After writing the book, Grisham became involved in work with the Innocence Project, and he soon realized the case was entirely typical. Unsound forensics is a national problem. "It is time to clean up the bad science," Grisham told the lawmakers, mincing no words where faulty science is rampant in American courtrooms. He called for urgent federal action to put a stop to the problem, including involvement of federal agencies to set binding scientific standards. We should not have 50 different definitions of science in each of the 50 states, he pointed out. During the hearing, Senator Amy Klobuchar asked, "Mr. Grisham, have you seen any ideas for books in this discussion here? Maybe you could do a thriller on the very slow process to get things done?" Grisham answered, "Well, everything is fair game for a book." Almost a decade later, very little has been done, and the federal government has not responded as he urged Congress to do. The hearings on the Senate floor never would have happened, though, if a prominent scientific group had not issued a report in 2009 that detailed the flaws in much of the forensics used in our courts. In this chapter, I tell the story of how this crisis became the subject of such urgent concern, and then the aftermath of the report that exposed a national crisis in forensics. The Judge and the National Academy. Judge Harry Edwards, a prominent federal appellate judge, had no preconceived views about the forensic science community. He had served on the law faculties at University of Michigan Law School and Harvard Law School, before serving on the board and then as chairman of Amtrak. He was appointed as a judge on the United States Court of Appeals for the District of Columbia and served as the chief judge. During his distinguished career, hearing appeals in complex and high-profile criminal matters, he simply assumed, like most judges, that forensic science was well-grounded in scientific methodology. He had written important judicial opinions and books, including on judicial process, legal ethics, and labor arbitration. But forensic science had never been a focus. Willing to take on a new challenge, Judge Edwards agreed to co-chair a National Academy of Sciences committee that would study the challenges facing forensics. The judge soon realized that he had much to learn about the state of forensics and the aggressive defenders of the status quo. At that time, I had similarly unformed views of forensics, and like most lawyers, I did not have a science background. As a young lawyer, starting in 2002, I had worked on cases of DNA exonerees. In a Staten Island case, I toured the DNA unit of the New York Medical Examiner's Lab to understand each step in the testing process that ultimately set our client free. The case involved a bite mark comparison too, where the dentist had claimed our client's teeth were consistent with a bite on the victim. However, as part of the effort to obtain compensation for the exoneree, the focus was on the DNA test later done on the swabs from that bite. As a civil rights lawyer, I had never carefully looked at the work done by bite mark, fingerprint, or hair comparison experts. Like Judge Edwards, I assumed most of it was good science. In 2009, now a law professor, I read stacks of criminal trial transcripts in the quiet comfort of my office. It is uncanny, in hindsight, that one of those records was from Keith Harward's trial. I coincidentally came across his case because of a call from the group Judge Edwards chaired. In 2008, I had just published my first article studying the cases of the first 200 people exonerated by DNA testing. Anne-Marie Mazza, Senior Director at the National Academy of Sciences, NAS, called me and described a committee of eminent scientists and lawyers who had gathered to examine the state of forensics. They wanted to learn about the role forensic testimony played in the cases of DNA exonerees. To study that question would require a massive research project. One would have to locate paper records of trial transcripts, read them, code them, and analyze the data. I was not sure I could take on a project that large. Before agreeing to produce the first-ever report on the role forensics played in DNA exonerations, I called my friend Peter Neufeld, co-founder of the Innocence Project. My first question to him: what is the National Academy of Sciences exactly? The National Academy of Sciences, established by an act of Congress, signed by President Abraham Lincoln in 1863, is the leading organization of distinguished scholars in the sciences in the United States. It did not take long for Peter to explain that this was an august body and that this committee was engaged in a crucially important undertaking. Membership in the NAS is considered to be one of the highest honors that a scientist can receive. 190 of the NAS members have received Nobel Prizes. The committee formed to study forensic science had 17 members, including several forensic scientists, leading research scientists, and prominent lawyers and judges. No one on the committee had ever criticized forensic methods in the past. In fact, 6 members had very close ties to forensic science. After hearing more about just how important this work was, I agreed to work with Peter to collect more information about what went wrong in DNA exoneree trials. We had just a few months to prepare an exhaustive report. Meanwhile, the committee's larger work would uncover a national crisis. The DNA Revolution. When DNA testing arrived on the scene in the 1990s, it put all of forensics to the test. Before DNA testing became common, blood typing could sometimes be done in criminal cases, and it could readily exclude a suspect. If the blood at the crime scene was type A, then it definitely could not have been left by a person who secretes type O blood. Since many people share the common A, B, and O blood types, this was not often very useful evidence to include a suspect. An expert might be able to say that a person was among a group of 30 or 40% of the population that shared that blood type. Also, you needed quite a bit of blood or biological material to do blood typing. Most crime scenes did not have that quantity of material. Such testing was particularly useful in sexual assault cases, or in cases where the culprit left a piece of saliva or sweat or blood-stained clothing. DNA revolutionized forensic testing, but not right away. DNA was first identified in the 1860s. In 1953, James Watson and Francis Crick announced their discovery of the double helix chemical structure of DNA. DNA resembles a twisted ladder, with the sides of the latter made up of alternating sugar and phosphate groups, and with the middle rungs of the strand containing just 4 bases: adenine, A, guanine, G, cytosine, C, and thymine, T. The patterns in those base pairs encode the instructions that DNA provides to our cells. Many thousands of those pairs may make up a single gene. Since its discovery, researchers have intensively studied the makeup of the human genome. DNA testing methods were developed in research laboratories, unlike much of forensic science, which was developed by police crime labs or assorted freelancers, like the dentists in Harvard's case. In the 1980s, British geneticist Sir Alec Jeffreys pioneered the genetics research that made DNA testing possible in criminal cases by focusing on short repetitive sequences within the genome that are highly variable between individuals. Scientists like Jeffreys developed ways to isolate those sequences. While nuclear DNA contains many genes that do important work, providing instructions for how to encode proteins, geneticists found that the vast majority of the sequences is non-coding, sometimes called junk DNA, although scientists are beginning to learn more about some purposes that it can serve. Early DNA testing required large quantities of biological material. However, it was not commonly used in criminal cases. The DNA revolution for criminal cases occurred by the mid-1990s, when a new technique called short tandem repeat, STR testing, was developed. The key words are short and repeat. The entire DNA strand is 6 feet in length, yet each chromosome is packaged incredibly tightly in the nucleus of a cell, so it is only a few microns, or 1/1,000,000th of a meter wide. The STR technique tests short portions of the strand, which means that very small samples of DNA can be tested. The repeat part refers to the fact that those portions repeat a number of times, which vary widely from person to person. A second feature of this genetics revolution was polymerase chain reaction, PCR technology. Scientists developed enzymes that make copies of copies of copies in a chain reaction that can produce billions of copies of the sample. Scientists developed rapid and largely computerized analysis of that genetic material as well. These STR and PCR tests were standardized in the 1990s, and today they test over 20 portions of the DNA strand to produce a DNA profile. Unlike traditional forensics, DNA testing relies on objective statistics. Researchers develop data on populations of people and the frequency of certain genetic characteristics, called alleles, that vary a great deal. The results of a DNA test do not provide a match, but rather a probability. Using population statistics, scientists can determine whether one person in many millions, or even billions or trillions, could be expected to randomly match a particular DNA profile. In contrast, there are no population statistics for characteristics observed on bite marks, hair, fingerprints, or other objects like firearms and tool marks. We have no idea how common or rare the details are that examiners rely upon for those forensics. Companies began to market DNA technology to law enforcement, promoting DNA fingerprinting as a way to get conclusive results in only one test. The FBI quickly took on a leading role, choosing its own protocols and building a new DNA analysis unit. The FBI became the go-to lab for DNA testing, although by the mid-1990s, more state and local labs began to test DNA as well. In 1993, Congress enacted legislation authorizing the FBI to assemble DNA samples in a national databank, the Combined DNA Index System, CODIS. Judges quickly ruled DNA evidence admissible evidence in criminal cases, although not without a fight. In its infancy, DNA experts sometimes would testify that DNA was a match, where they used probabilities that were not based on careful calculations. In response, defense lawyers called serious researchers in academic molecular biology and genetics to testify. High-profile disputes about the lack of transparency and potential subjectivity of DNA methods led to the DNA wars of the 1990s. The National Academy of Sciences convened leading scientists to study the problem and settle on clear standards for forensic use of DNA testing. The impact of DNA on the criminal justice system quickly became apparent. The first Innocence projects took off. Lawyers like Barry Scheck and Peter Neufeld, who founded the Innocence Project in New York City, realized that time is on the side of DNA testing. DNA material can remain intact for years, so decades after a conviction, evidence could be retested. In hundreds of cases, 370 and counting, post-conviction DNA testing has proved the innocence of convicts. The Innocence Project leads in the use of DNA testing to exonerate. But their work has also sparked a national and global network of Innocence projects. Each year at the annual Innocence Network Conference, exonerees take the stage to celebrate their hard-won freedom. And while the early conferences were smaller, today it can take more than an hour just to introduce all of the exonerees attending. These people spent, on average, over 15 years in prison. For years, prosecutors and judges insisted on their guilt, and often it took years of fighting in the courts for lawyers to secure their freedom, although more recently some prosecutors have formed conviction integrity units and have themselves reinvestigated cases and secured exonerations. Much can be learned from studying these miscarriages of justice. My first book analyzed the cases of the first 250 DNA exonerees in detail. I might never have begun that work had Anne-Marie Mazza not called from the NAS, asking me to report on the role that forensic science played in exoneration cases. The U.S. Congress had asked scientists at the National Academy of Sciences to re-examine forensic science, in part because the steady drumbeat of DNA exonerations increasingly called forensics into doubt. While great improvements in forensics had been made, and crime labs increasingly had larger and dedicated professional staffs, the scientific underpinnings of entire disciplines had been called into question. Moreover, crime labs were facing quality control scandals. For example, in Chapter 11, I will describe an audit of the Houston Crime Laboratory that uncovered errors in hundreds of cases, including in the DNA unit, and ultimately shut down the lab. As cases were reopened, labs investigated, and scandals proliferated, lawmakers called for a serious scientific review. Flawed forensics in DNA exoneration cases. Forensic science itself needed an autopsy to uncover what had gone wrong with the forensics in so many innocent people's trials. Fortunately, at the time that the committee reached out, the Innocence Project had recently digitally scanned its files. Peter Neufeld and I decided to work together on the post-mortem, an analysis of the trial testimony of forensic analysts in DNA exonerees cases. We planned to read and code these trials and then report our findings to the NAS committee. We also wanted to see whether the forensic expert testimony in these innocent people's cases was typical or not. That was why I was reading randomly pulled trials from Virginia, like Keith Harward's, as well as criminal trials from several other states. We soon saw a clear pattern. While hundreds of innocent people had been freed by DNA technology, most had been convicted in the first place based on traditional forensic evidence, like hair, bite mark, and even fingerprint comparison. Peter Neufeld and I found that in the majority of these innocence cases, the forensic evidence was not just in error. Analysts had overstated the evidence in court and given the jury an exaggerated picture. We found the same flawed testimony in the random trials that we picked. This suggested forensic misconduct was an ingrained feature of serious criminal trials. We told the committee that flawed forensic testimony in the DNA exoneree cases was extensive, and it was just the beginning of a deep and disturbing problem. Author John Grisham, commenting on these findings, noted most DNA exonerees were convicted by forensics, much of which was flawed, unreliable, exaggerated, or sometimes outright fabricated. Our courts, Grisham said, have been flooded with an avalanche of unreliable, even atrocious, science. Today, 370 people have been exonerated by post-conviction DNA testing in the United States. The same troubling figures that I relayed to the National Academy of Sciences still hold true in this larger and still growing group of innocent people. Of those 370 exonerees, at least 250 had forensic evidence in their cases. Some had forensic tests results that should have helped them prove innocence at the time of their conviction. 52 of those exonerees, for example, had forensic evidence that outright excluded them and pointed to their innocence at the time they were convicted. In 30 cases, though, this forensic evidence of innocence, including DNA or fingerprint or other comparisons, was concealed from the defense. However, far more had prosecutors present forensic evidence that contributed to their false convictions. In my latest update of the data, I found 114 of the 250 had invalid presentation of forensic science at the time that they were convicted. 20 more had vague testimony presented that did not explain the conclusions reached. In 31 cases, we know of serious errors made during the analysis. Thus, well over half of these exonerees were freed by DNA testing, but they were wrongly convicted in the first place based on flawed forensics. Resisting science. Leading scientists had never scrutinized most of the forensic disciplines, the non-DNA forensics that really needed it, apart from two very critical NAS reports on voice comparison and bullet lead comparison evidence. Fingerprints have been admitted in criminal cases in the United States since 1911, and handwriting and firearms comparisons have been used for a century or more. Other techniques were more recent, like bite mark and bullet lead comparisons. The FBI stopped using bullet lead comparison method after the NAS report debunked it. Nor were the problems with forensics unknown, as DNA exonerations mounted. Congress poured hundreds of millions of dollars into reducing DNA backlogs, creating DNA databanks, and expanding crime lab capabilities in DNA. The other forensics took a back seat. Law enforcement tried to stymie efforts by scientists to study non-DNA forensics. In fact, the National Academy of Sciences planned to study non-DNA forensics earlier, in 2002. In a revealing editorial, the editor-in-chief of the scientific journal Science asked if forensic science was an oxymoron. He described how the Department of Justice and Department of Defense insisted on rights of review before the NAS could publish their findings. Rightly concerned with this imposition on scientific integrity, the NAS refused to review forensics under such conditions. Finally, in 2005, Congress appropriated funding for a major NAS effort, and the committee began its work in fall 2006. The committee held meetings over many months. They began by inviting the entire forensics community to submit research and papers. The experience forever changed the views of these scientists and lawyers, as they became exposed to problems that they had never known to have existed. At the committee hearings, an expert fingerprint analyst was asked about the extent of research on how well a fingerprint examiner can make a conclusion about a smudged or partial print. The expert admitted, research has yet to be done. One of my friends on the committee told me how you could practically hear a pin drop in the room when he made this admission. Another forensic expert explained microscopic hair analysis to the committee. Judge Harry Edwards asked the expert, "If your daughter was falsely accused of a felony, and the only evidence against her was a microscopic hair sample, how would you feel?" He responded that he would be very concerned. Judge Edwards was troubled to hear that the expert was essentially conceding that a person might be wrongfully convicted if hair evidence was the basis for a prosecution. In another telling exchange, Peter Neufeld and I told the committee about the case of Neil Miller, a DNA exoneree. At Miller's trial, the forensic analyst had described how the blood type from the evidence collected from the rape victim was consistent with Miller's blood type. He testified that approximately 45% of the population had type O blood, like Miller did. The analyst never explained that 100% of all men could match the evidence. At the time, they could not separate or quantify the fluid from a male contributor to a sample. The victim's fluids could mask any contribution from the culprit. It was obvious to any competent expert that if nothing different from the victim's blood type was observed, then no conclusions could be reached at all about the culprit's blood type. Miller was convicted based on flawed forensic testimony, and yet this was not a junk science. ABO blood typing is highly reliable and based on sound population statistics. Instead, the problem was that the analyst gave misleading statistics to the jury. Over 50 DNA exonerees were convicted based on misleading testimony regarding traditional blood typing. After reading this testimony to the committee, a crime lab director forcefully objected. He said that there was nothing unethical about the testimony that led to Miller's wrongful conviction, because it is the job of defense lawyers to bring out any problems with testimony, and it is not the job of the lab analyst to offer the right statistics. I could tell from their expressions that many in the room were aghast that the director of a lab viewed highly misleading forensic testimony as entirely appropriate. What made the moment still more telling was our next slide. In another case, that of DNA exoneree Marvin Mitchell, the same analyst testified, and this time, where it helped the prosecution's case, he explained why blood typing could be such a limited form of evidence. In Mitchell's case, the blood type observed matched the victim but not the defendant. This time, the very same lab analyst who testified in Neil Miller's case explained that the stain might have been too diluted to pick up Mr. Mitchell's blood type. This lab analyst was selectively presenting misleading statistics to help the prosecutors to convict defendants, at least two of whom later turned out to be innocent. Scientists take a stand. The forensic science community could talk about nothing else in the months before the National Academy of Sciences Committee released its report. At the National Conference of the American Academy of Forensic Sciences, which began on the day the report came out, lab analysts were asking, "What does it say? Have you read it?" On February 16, 2009, the report Strengthening Forensic Science in the United States, A Path Forward was published. The response by many forensic analysts when they read the report was surprise, disbelief, resentment, anger, and indifference. The report was not fair, and ivory tower academics don't get us, many responded. After all, one after another, forensic practitioners had told the committee they were positive that the work that they were doing was fine. The committee produced a book-length 300-page report with 13 main recommendations. The committee comprehensively reviewed the state of the research across a host of forensic techniques. Perhaps the most quoted portion of the report concluded that much of forensic evidence used in criminal trials is without any meaningful scientific validation. The committee described major problems in forensics, including where faulty forensic science led to wrongful convictions. They described the need for more research. Many types of forensics work simply did not have reliable scientific methods or foundations. They noted that the judges and courts will not cure the infirmities of forensic science. The problems are too ingrained, and judges have been utterly ineffective. That was a damning critique from a committee co-chaired by a prominent federal judge. Identifying major gaps in the research was a big part of the committee's job. But so was identifying solutions. They called for a national response to a national crisis. Congress needed to create a National Institute of Forensic Science to develop standards and ensure that forensic science work is done in a truly scientific manner. Thousands of media outlets reported on the findings. You can see the complete list of stories on the NAS website. And you just keep scrolling and scrolling. Past stories in the New York Times, Nature, Science, and more. Television shows from Nova to NCIS to John Oliver's Last Week Tonight have covered the report. Commentators called the report a blockbuster and a watershed. Congress held hearings, and legislation was introduced to regulate forensics along the lines that the NAS proposed. None of that legislation passed. The U.S. Supreme Court promptly cited the report, with Justice Antonin Scalia noting that forensics have serious deficiencies and are not immune from the risk of manipulation. The President appointed a committee on forensic science. Traditional forensics, however, had powerful defenders. One prosecutor, testifying before the Senate, called the report an agenda-driven attack on well-founded investigative techniques. Then Senator Jeff Sessions said, "I don't think we should suggest that those proven scientific principles that we've been using for decades are somehow uncertain." The dentists respond. The dentists that compare bite marks unwittingly give us a test case for how our system responds to a ringing statement from the scientific community that techniques used for decades lack support. The National Academy of Sciences report discussed bite mark comparisons specifically, stating there was no evidence of an existing basis for using bite marks to identify an individual. The committee concluded that scientific research must be done to know whether a bite mark comparison can ever provide probative value. The forensic dentists were listening. In response, in 2009, the American Board of Forensic Odontology changed its rules for its members. They now stated that terms such as unconditional identification of a perpetrator or identification without doubt are not sanctioned as final conclusions in an open population case. They changed their wording. The White House Report. Eric Lander, a mathematician and geneticist, founder of the Broad Institute of Harvard and MIT, and a leader of the Human Genome Project that led the way in mapping our DNA, met with leadership at the Department of Justice. These federal prosecutors told him they needed a grace period. Prosecutors would be in a lot of trouble if scientists called into question a host of forensic methods. Yet this was over 6 years after the NAS report, and foundational work had not been done on many forensic techniques. Very little had changed, except perhaps for the wording of some forensic conclusions. In fall 2015, the White House had convened a committee of the President's Council of Advisors on Science and Technology, PCAST, which focused more deeply on problems in a handful of important forensic disciplines: analysis of DNA mixtures, firearms comparisons, fingerprint comparisons, and bite mark comparisons. Like the NAS committee, this group brought together prominent scientists, including Lander. "We concluded it was not within our roles as scientists to grant grace periods," Lander recalled. They went ahead and released their report, which was very forceful. They stated unequivocally that unscientific forensics should no longer be used. There should be no grace periods, no extensions. Such forensics should not be used until real studies were done to show they are reliable. The scientists reviewed thousands of studies assessing the reliability of forensic methods, but found only a handful that had been adequately designed. An adequately designed study must include real tests of the technique, with large samples, independent researchers running the study, and open data that others can reproduce. In other words, they must be real scientific studies. For some techniques, like fingerprints, PCAST said there had been a minimally adequate number of real studies, at least 2 done on the technique. PCAST described undocumented error rates and how even for methods like fingerprinting, error rates were far higher than jurors and lawyers might assume. People needed to be fully informed about these error rates in criminal cases. Techniques like firearms and bite mark comparisons were different. Adequate studies had not been done. The PCAST report found, bite mark analysis does not meet the scientific standards for foundational validity and is far from meeting such standards, they said. To the contrary, available scientific evidence strongly suggests that examiners cannot consistently agree on whether an injury is a human bite mark with reasonable accuracy. They concluded that techniques like bite mark and firearms comparisons should not be used in court. The FBI promptly responded that it disagreed with many of the scientific assertions and conclusions in the report, and that it failed to mention numerous published research studies that were sufficiently well designed. In response, PCAST invited the FBI or any others to submit any published studies that they had somehow missed that might speak to the reliability of these forensic methods. The DOJ concluded that there were in fact no additional studies for PCAST to consider. The loudest protests came from prosecutors, who roundly criticized the report. The Department of Justice said that they would ignore the report outright and would continue to use forensics the way they always had. Then Attorney General Loretta Lynch explained that the DOJ was confident that, when used properly, forensic science evidence helps juries identify the guilty and clear the innocent. At a law school event on forensics, the DOJ's forensic science spokesperson, Ted Hunt, argued that a flexible and holistic test should be used to decide what is valid science. Lander responded that requiring studies be done with large samples, independent research supervision, and reproducibility is not too narrow a definition. It is what is called science. The National District Attorneys Association accused PCAST of pervasive bias and called the report scientifically irresponsible. This was not because of any flaws in the recommendations, but because adopting them would have a devastating effect on law enforcement. The leading organization of fingerprint examiners said it stood behind the reliability of fingerprint evidence. In 2016, the Texas Forensic Science Commission announced a moratorium on the use of bite mark evidence in the state. No other state has followed their lead and taken that step. This story is like the movie Groundhog Day, in which Bill Murray relives the same day over and over again. We have seen the same plot line for other flawed forensics techniques, including some that have been discredited as far too unreliable to be used in court. And yet judges still let experts testify about the techniques in court. The NAS issued a scathing report concerning voice comparison in 1979. The FBI, to its credit, stopped using the technique in response, but state courts continued to let it in as if nothing had changed, including in the case of an innocent man, David Sean Pope, who was later exonerated by DNA testing. For decades, the FBI used a technique called comparative bullet lead analysis to link bullets to a particular box of ammunition in evidence. The NAS finally reviewed the research, and in 2004 wrote a report concluding that one cannot support any statement that a crime bullet came from a particular box of ammunition. People had been convicted, even executed, in cases relying on this bogus bullet matching technique. The FBI initially claimed that this technique was still usable. Over a year later, the FBI finally discontinued the technique, explaining that it had become too costly to maintain the equipment. Statisticians, Drs. Karen Kafadar and Clifford Spiegelman, examined an FBI database that it had refused to share with the NAS, but which it claimed had supported this technique. They found the database was riddled with errors and inconsistencies, hidden and flawed data, stubborn refusal to follow the science, and grave consequences. This has been the story of forensic evidence, time and time again. A response to critical scientific reports, however, continues to be defensiveness, denial, and anger. As then Deputy Attorney General Rod Rosenstein put it in a speech in 2018, forensic science is under attack, and critics have an erroneously narrow view of forensics because they expect it to all be statistics and automated. That is not so. Experts do not need to be replaced by computers. Human experts can reach accurate judgments, but we need real evidence that the methods they use are in fact reliable. Good science. Stubborn resistance to criticism and hostility to scientific research is the very antithesis of good science. As the New England Journal of Medicine and other leading medical journals put it in a brief to the U.S. Supreme Court, the concept of good science is simple. It is the scientific community's system of quality control, which protects us all from unsubstantiated analysis. Any proposition must undergo a rigorous trilogy of publication, replication, and verification before it is relied upon. What leading scientists keep telling us is that, apart from the DNA area, no forensic techniques have undergone sufficiently rigorous testing. Error rates have been unknown, as the next chapter will describe. Simply put, we need to bring good science to forensics.